Welcome to the PubMed Daily Review for March 22 SD. Today we scanned 1953 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5. Living cellular biocomputing designs. Living cellular systems are being engineered for complex computation and pattern formation. This cluster covers 321 papers. Representative work includes genetic designs for stochastic and probabilistic biocomputing, why cellular computations challenge our design principles, and Turing patterns with cellular computers. Trend 2 of 5. Cancer immunotherapy and gene regulation. These papers explore how gene regulation impacts cancer development and immunotherapy effectiveness. This cluster covers 246 papers. Representative work includes erratum for the research article, cooperative role of distinctive TP53 and PTN combined loss in the peripheral T-cell lymphoma, GATA3 molecular subgroup by W.G. Lone et al., erratum for the research article, and 1 is a critical regulator of R-loop dynamics and a target to overcome endocrine resistance by Z. Lee et al., and caspase 3-7 deficiency results in enhanced intestinal inflammation and reduced tumorigenesis. Trend 3 of 5. Clinical management and health access. These papers explore optimizing patient care and improving healthcare accessibility through innovative clinical strategies and technological integration. This cluster covers 517 papers. Representative work includes navigating the diagnosis and management of papillary muscle rupture following inferior myocardial infarction, a case report of general anesthesia for abdominal surgery in severe cavitary tuberculosis, strategies to minimize barotrauma, and long-term clinical follow-up of intracranial aneurysms treated with flow diverter devices. A bicentric retrospective study. Trend 4 of 5. Protein structure and genetic analysis. This trend explores how protein structure and genetic analysis inform biological function and adaptation. This cluster covers 485 papers. Representative work includes large-scale clustering of AlphaFold 2 3D models, shines light on the structure and function of proteins. Geneta, a network-based tool for the analysis of complex genetic designs, and parameter evolvability in gene expression models drives phenotypic adaptation. Trend 5 of 5. Brain imaging and mental health. These papers explore brain structure, function, and molecular mechanisms related to mental health and neurological disorders. This cluster covers 384 papers. Representative work includes the relationship between emotional dysregulation and HIV acquisition risk behaviors and intimate partner violence perpetration among young men in rural areas and urban informal settlements in South Africa. Structural and functional asymmetry of the neonatal cerebral cortex and supervised phenotype discovery from multimodal brain imaging. Up next, our first paper out of 5. An agentic AI system enhances clinical detection of immunotherapy toxicities, a multi-phase validation study by Jack Gallifant and Danielle S. Bitterman. Let's break down its key contributions. Immune checkpoint inhibitors are revolutionizing cancer treatment, but they can cause significant side effect called immune-related adverse events, or IRAs. Identifying these toxicities is crucial for patient management, but it's currently a manual, time-consuming, and often inconsistent process. This paper investigates whether a new AI system can help. The researchers developed an AI system designed to read clinical notes and identify specific immunotherapy toxicities. They tested this system in multiple phases. First, retrospectively on a set of notes, then prospectively in a real-world setting, and finally, in a randomized study comparing AI-assisted annotation to manual review by clinical staff. The AI system performed exceptionally well, achieving a high F1 score of 0.92 for detection in retrospective analysis and 0.72 to 0.79 in prospective deployment. Crucially, when used by clinical staff, the AI assistant reduced annotation time by 40% and increased accuracy, with an odds ratio of 1.45 for complete matches. It also substantially improved agreement between annotators. This study shows that AI, when combined with human oversight, can be a powerful tool for managing immunotherapy side effects. It's not only accurate, but also cost-effective and speeds up a critical but tedious task. This technology holds great promise for improving how we monitor patients on these life-saving therapies and could accelerate clinical research. Up next, our second paper out of 5. Machine learning-informed Nano co-assembly inhibits fibroblast activation protein and improves drug delivery in fibrotic tissue by Zhihua Liu and Xiaofeng Yi from Drug Research Program. Division of Pharmaceutical Chemistry and Technology, Faculty of Pharmacy, University of Helsinki, Helsinki, Finland. Let's break down its key contributions. Delivering drugs effectively with nanoparticles is a major hurdle in medicine. Current methods are often complicated to make and don't reach their targets well, especially in fibrotic tissues. These fibrotic environments create barriers that prevent drugs from getting where they need to go. We investigated a new strategy using a special molecule to improve nanoparticle design and drug delivery. We screened over 800 compounds to find a suitable excipient, which we identified as SP-13786. This compound was then used to create stable nanoparticles, called SCAN, through a simple coprecipitation process. We employed advanced machine learning techniques to understand the principles behind this co-assembly. Finally, we tested the effectiveness of these SCAN nanoparticles in FAP-positive cells and in animal models of fibrosis. Our results were very promising. We found that using SP-13786 as an excipient broadly enhanced the drug loading capacity of the nanoparticles. This improved bioavailability directly correlated with superior therapeutic outcomes in our disease models. The SCAN nanoparticles demonstrated enhanced drug delivery in FAP-positive cells and effectively attenuated fibrosis-induced barriers, leading to better drug accumulation in fibrotic tissue. This study establishes SP-13786 as a valuable tool for creating advanced nanotherapeutics. The combination of simplified preparation, enhanced drug delivery, and mechanistic understanding for machine learning is a significant step forward. This platform shows great promise for treating a wide range of fibrotic diseases where current drug delivery methods are insufficient. Up next, our third paper out of 5, population-scale burden analysis of rare damaging coding variants identifies novel risk genes for Alzheimer's disease and Parkinson's disease by Jan Luguen and Michael D. Breisius. Let's break down its key contributions. Alzheimer's and Parkinson's diseases are devastating conditions with a strong genetic link. While we know some genes are involved, the impact of rare, damaging genetic variations across large populations remains a mystery. Understanding these rare variants is crucial because they might point us to entirely new genes that play a role in these diseases, opening up new avenues for research and treatment. To tackle this question, researchers analyzed whole genome sequencing data from massive population biobanks and specific patient groups. They focused on rare variants that are either loss of function or predicted to be damaging missense mutations. By using proxy phenotypes, they aim to boost their ability to detect genetic signals for these late-onset neurodegenerative conditions. This approach allowed them to systematically test genes for an increased burden of these rare variants in individuals with Alzheimer's and Parkinson's. The study successfully confirmed known rare variant associations in genes like PSEN1 for Alzheimer's and LRRK2 for Parkinson's. Excitingly, they also uncovered several novel genes associated with increased risk for both conditions, including IMPA2 and SYNE1 for Alzheimer's, and ANKRD27 and CCL7 for Parkinson's. The strongest new signal pointed to ANKRD27, suggesting its variants are particularly important in Parkinson's disease pathogenesis. This research highlights the immense value of large-scale genetic studies in uncovering the rare variants that contribute to complex diseases like Alzheimer's and Parkinson's. The identification of novel risk genes provides exciting new avenues for understanding the underlying biology of these conditions. Further investigation into the precise functions of these newly discovered genes is now warranted, potentially paving the way for future therapeutic strategies. Up next, our fourth paper out of 5, Overcoming T-cell tolerance to tumor self-antigens through catch bond engineering by Xiaojing Chun and K. Christopher Garcia from Department of Molecular and Cellular Physiology, Stanford University School of Medicine, Stanford, California, 94305, USA. Let's break down its key contributions. Tumors often express self-antigens that T-cells are trained to ignore due to central tolerance. This makes it difficult for our immune system to effectively fight cancer. We investigated a new way to engineer T-cells to overcome this tolerance and become potent anti-tumor agents. We focused on engineering T-cell receptors, or TCRs, to overcome tolerance. By identifying and modifying a specific catch-bond hotspot within the TCR, we aim to increase its interaction time with tumor antigens. This involved advanced biophysical techniques and structural analysis to understand the mechanism. The results were striking. T cells engineered with a modified TCR showed vastly superior expansion within the tumor microenvironment. Crucially, these cells demonstrated potent effector phenotypes and were able to effectively eliminate tumors, demonstrating a significant breakthrough. This study presents a powerful new strategy for cancer immunotherapy by engineering T cells to overcome self-tolerance. This catch-bond engineering approach holds great promise for developing more effective T cell-based cancer therapies. Future work will focus on translating these exciting findings into the clinic. Up next, our fifth paper out of 5, fluid-derived organoids from pleural effusion and ascites. Emerging models for drug resistance and personalized oncology by Yongyang Chun and Wenmei Su from Department of Pulmonary Oncology, Affiliated Hospital of Guangdong Medical University, Zhanjiang, 524001, China. Let's break down its key contributions. Advanced cancers often lead to fluid buildup in the chest or abdomen, known as malignant pleural effusion and ascites. These conditions signal that the cancer is progressing and becoming resistant to treatments. While traditional tissue samples are useful, they can be hard to get and might not show how the cancer changes over time. That's where fluid-derived organoids, or FDOs, come in. These are tiny, lab-grown models made from cancer cells found in these effusions, offering a promising new way to study drug resistance and personalize cancer care. This review focuses on fluid-derived organoids, or FDOs, which are grown from cancer cells found in malignant effusions. The process involves collecting fluid samples, isolating tumor cells, and culturing them in a 3D matrix with specific nutrients. These FDOs are designed to mimic the patient's tumor, retaining its genetic makeup and characteristics. The key idea is that these models can accurately reflect how a patient's cancer behaves, including its resistance to drugs, and can even capture the complex interactions within the tumor's microenvironment. The studies reviewed show promising results for fluid-derived organoids. Establishment rates for organoids from ascites are quite high, between 60% and 90%, while those from pleural effusions are a bit lower, ranging from 30% to 60%. Importantly, these organoids have been shown to maintain the genetic features of the original tumors. Even more exciting, the drug responses observed in these organoids in the lab often match what patients experience in the clinic, suggesting they can predict treatment effectiveness. So, what does this mean for patients? Fluid-derived organoids offer a powerful, less invasive way to understand why cancers become resistant to treatment and to test potential therapies. While there are still hurdles to overcome, like standardizing how these organoids are grown and ensuring they fully represent the complex tumor environment, they hold immense potential. With further development, FDOs could become a crucial tool in selecting the most effective treatments for individual patients with advanced cancers.